Today we are trying to talk about AI and how the AI accelerated the bioprocess. We're trying to make the bioprocess fast and better. I'm Ken from Great Bay Bio. We into the site-specific integration platform for cell line development. So we have several advantages. The first thing is very fast. It only takes like 1.5 months to get the RCB, which is insanely fast, I have to say. And the second, we can have some product from the stable cellular cell line in the early stage, and then we can decide or we can choose the best one. And the third one is for the BSAB or some difficult to express protein. We can do that very good. See what we can do. So this shows a very traditional way we call the random insertion for cell line development. You can see that when we have a GUI, we can't control it. We just use the electrophoresis for it to inside into the genome of chose cell, and we get a pool of every kind of cell. It may insert into a good spot, maybe into the bad spot, and we don't know. So we have to do a lot of screening to get the best one. The other rationale or other choice is for site-specific integration. That means we find the best spot for protein expression in CHO cell, and then every time we do the cell line development, we put the GUI into the site, which we call the hotspot. Then we will get a very uniform cell line, and we need no screening work anymore. We just select it or pick it up, that's all. So here we are doing this work. We find the hotspot in the CHO cell, and we mark that with the GFP, and when we do the transfection, the GFP will be substituted by the GUI in the homologous recombinase manner, and then we will get what we want. And so the most important thing here is for the hotspot. How can we find it? Here we use AI to do the work. So generally, the face recognition for humankind, we can distinguish different humans using their face or using visual recognition. And for cells, that's the same. We can distinguish the high producers and low producers from its appearance or from its morphology. So that is what we are trying to do. We correlated the morphology with the protein titer. And then we have the algorithm. And then we can do things to pick up the highest expressed cell from a cell pool. And for this, we have to do more than 100 million cells to pick the best one. And this is show how we do. We will use a microscope to test, to check the morphology, and we will mark the high producers with numbers. And then we will use a self-designed automated robot to pick the cell up and into the 96-well plate. And it is a high producer with the gene, or the GFP gene was inserted into the right spot. So we will sequence it and to find the hotspot in it. So the following work is quite easy. So how we can do RCB in 1.5 months. So what we get as a host cell is the cell with the GFP. It has fluorescence of green. And when we have the GUI into it, we use electrophoresis. There will be two kinds of cell. One kind is a GFP cell, green, and the other one is no color cell or no fluorescence cell. And that is what we are trying to get because the GUI have substitute the GFP in these cells. So we are trying to find the non-GFP cell and to select it or pick it up. In this process, it is very important because all the non-fluorescent cells are created equal. So we don't need very high cell viability, we don't need very high transfection rate. In our experience, maybe 1% of the positive cells enough for us to pick it up. And also the viability, maybe 10% or something is good. So that could enable us to shorten the time after transfection. So always we need only 1 week for this work. That means after transfection, only 1 week later, we will do the cell selection. And also because we are trying to get the monoclone directly, so we can use VIPs or FACS to pick the cell up. The difference is for the GFP. For VIPs, it don't have the ability to pick the non-fluorescent cell only. So we have to pick more of the cells. And for FACS, we can select the non-fluorescent cell, so maybe less plate for us to do. Anyway, we have only to get 200 clones. That's enough for us to carry on the RCB construction or development. So the one way is we directly use the monocell for RCB development because it's already to be a monoclone, so you don't need any other things to do, just to let it grow. And the other thing is we can collect all the non-fluorescent cells to form a cell pool. That means we can, it may take only 1 month before we can get the cell pool, and you can get some products from the stable cell pool, and to have the attributes assessment or anything you like PD works. So all the process from the electrophoresis to the RCB development, it takes only 6 weeks. So it's because we bypassed the mini pool. Maybe you guys know that. We have to screen and screen from the mini pool. We don't need that. We pick the monoclone directly. And the second thing is we need only 7 days post-transfection because we don't need a very high cell viability or the transfection rate. And the third one is wide application. Because the hotspot is quite strong, we can express MAB, BSAB, or any kind of recombinant. So that is the process, and that is the result. So the first very important thing is stability. So we guarantee the stability because the site is stable. First, it is a single integration site. We only have one site, and we only use that site as our candidate hotspot. And also the site to express protein, its stability is predetermined because we screened a lot of candidates in the hotspot selection step. And also we have tested stability, both in the host cell and in our produced cell lines. So in the right one, you can see the different product after 90 days of culture, it maintained the titer of higher than 80%. And this is the appearance, and what about the genome scale? So first, the copy number maintained at around 1 because we have only one site, and also the insertion point or the hotspot remained at the designed hotspot as we use the next generation sequence for this test. And we can see from P0 to P18 that may take like 18 generations and like 60 days, it maintained at a very stable level. So we can know that the cell can stably produce what you like to produce in a very long time because it have only one hotspot, and that is the key. And the second most important thing is for the titer. And now we can see in the right part, in your left part, you can see the 3 different kinds of antibodies, IgG1, IgG4, and BSAB. Interestingly, we always can make titer higher than 5 grams per liter for the antibodies. And also we have tested some fusion proteins and also IVD proteins, means the antigens and some antibodies from different animal species. And also we can maintain at expression titer of higher than 3 grams per liter. So that is result. And especially for the BSAB, we think we can use the stable cell, the site-specific cell line development to solve the pain points. The first is titer, the second is correct pairing. So we tested different kinds of BSABs, and from the 2 chains to the 4 chains and to the 3 chains. And always we can get a very high titer, like 5 or even higher than 10. And even important, we can get a correct pairing rate of higher than 90%. So that is not normally happen. So the most important thing is we do the rational vector design. So we can see that we have 4 designs at least for different kinds of BSABs. What are these? Let's see, we have different tools for BSAB. The first tool is for the signal peptides. Signal peptide is really important in cell line development. It is 0 or 1 difference. So we have AI-generated signal peptides customized by the antibody we're trying to produce. And we also have some commercialized signal peptides. So when we're doing a BSAB, we use different combinations of signal peptides and try to find the best combination for them. So as a small conclusion for the AI-enabled site-specific cell line development, first is very quick. I think it is the most fastest way I can, as far as I know. And also, we have only minimum workload, only 200 clones in 96 well plate, and only 20 in 6 well plates. And the derivation may be less than 20% in titer, and nearly no difference in quality. And also, it is a very lean process. We need very little workload to develop the cell line, because we don't need any mini pour, and it only takes 7 days post-transfection. And also, we can guarantee the titer of MAB higher than 5 gram per liter, which is normal, and also BSAB for higher than 5 gram per liter. And also, we can produce antigen or other very difficult to express proteins higher than 1 gram per liter. We have also filed the IND for DMF to facilitate our IND filing.